Hello, my name is Aditi Srinivasan and I am a student at William Mason High School. Today I am presenting my research project linking PFAS exposure to high cholesterol prevalence in Ohio, evidence, analysis, and community impact. The intersection of environmental quality and public health is a critical modern concern. In this study, I investigated the relationship between trace chemical exposures in public drinking water systems and community-level cardiovascular risk factors, specifically hypercholesterolemia, across 27 municipal populations in Ohio. Today, I'm going to discuss the nature of these persistent contaminants, the multivariable linear regression model I built to evaluate their health impacts, and the implications these findings hold for environmental regulations and clinical practices. To understand this issue, we must first define PFAS. PFAS stands for per and polyfluoroalkyl substances. These synthetic organic molecules contain exceptionally strong carbon-fluorine bonds, making them highly resistant to heat, water, oil, and metabolic degradation. Because they do not break down easily in ecosystems or the human body, they are commonly referred to as forever chemicals. Due to these properties, PFAS has been heavily used in manufacturing since the mid-20th century. As shown on the slide, they are found in an enormous array of consumer products and industrial applications. They are the active components in firefighting foams used at military bases and airports. They line microwave popcorn bags and fast food packaging and are embedded in water-resistant clothing, stain-resistant furniture, non-stick cookware, cosmetics, and pesticides. Because of their ubiquity, these chemicals inevitably leach into our surrounding ecosystems during production, usage, and disposal. This widespread industrial use has led to severe environmental contamination, particularly in our drinking water infrastructure. Recent data indicates that PFAS is present in approximately 45% of all United States tap water supplies. These compounds enter the water cycle through industrial runoff, wastewater treatment discharges lacking specialized filtration, landfill leaching, and historical firefighting foam usage. Once inside groundwater or surface water systems, they travel long distances. While contamination is significantly concentrated in urban areas and near waste disposal sites, it also impacts rural private wells and protected public water supplies. This creates a continuous pathway for human chronic exposure through everyday water consumption. The environmental presence of PFAS is highly concerning because chronic exposure is linked to severe multi-system health risks. First, exposure is tied to reproductive and developmental issues, including increased maternal blood pressure, low infant birth weight, and developmental delays in children. Second, it carries oncological risks, showing an increased statistical likelihood of kidney and prostate cancers. Third, these chemicals cause immune and endocrine disruption, reducing vaccine effectiveness, lowering the body's ability to fight infections, and interfering with natural hormones. Finally, and central to this study, PFAS exposure induces metabolic changes, explicitly manifesting as increased serum cholesterol levels and a higher risk of thyroid disease and obesity. Is there a correlation between PFAS in drinking water and higher cholesterol in the human bloodstream? Biological research points to specific pathophysiological mechanisms. When ingested, PFAS compounds bind to proteins and accumulate in the liver, the primary organ for lipid metabolism. PFAS molecules alter liver lipid metabolism and induce hepatotoxicity. They disrupt nuclear receptors that regulate lipids, leading to reduced cholesterol excretion and altering how low-density lipoproteins are cleared from the blood. Additionally, endocrine disruption further aggravates this metabolic imbalance. Because elevated cholesterol is a major risk factor for cardiovascular disease, understanding this independent environmental contributor is vital for public health. In response to these risks, the regulatory landscape has recently shifted. The U.S. Environmental Protection Agency enforced a national standard setting maximum contaminant levels, or MCLs, in drinking water for several common PFAS variants. Specifically, the legal limit is set at a strict 4 parts per trillion for both PFOA and PFOS. Achieving these low thresholds requires massive municipal efforts. Standard treatment methods are insufficient. Water systems must utilize advanced engineering technologies such as granular activated carbon, ion exchange resins, and high-pressure membrane filtration, while residential areas often rely on reverse osmosis to filter out these microcontaminants. While national data such as this map from the Environmental Working Group illustrates widespread contamination, I wanted to analyze the localized health consequences within Ohio. The purpose of my study was to investigate the relationship between three prominent PFOS compounds, PFOS, PFOA, and PFBS, and the prevalence of high cholesterol among adults across various Ohio cities. My primary research objective was to determine if local drinking water PFOS concentrations serve as a significant predictor for community-level hypercholesterolemia. Crucially, I wanted to find out if environmental exposure acts as an independent contributor to cardiovascular risk factors when traditional lifestyle variables are accounted for. To execute this investigation, I analyzed data from 27 cities in Ohio, selected based on availability of public water monitoring reports and matching community health statistics. The target population consisted of adults aged 18 and older served by municipal water systems. Data collection integrated three distinct sources. Environmental exposure metrics were obtained from public drinking water reports, tracking measured concentrations of PFOS, PFOA, and PFBS. Cholesterol prevalence statistics were sourced from the CDC Places database, which records the percentage of adults diagnosed with elevated cholesterol per locality. And then finally, demographic and lifestyle confounders, specifically median age, obesity rates, and smoking prevalence, were obtained from the Ohio Department of Health to serve as mathematical controls. Because cholesterol levels are heavily influenced by lifestyle and biology, a simple correlation would be insufficient. To isolate the true environmental impact, I developed a multivariable linear regression model. This approach allowed me to evaluate the predictive power of environmental exposure while controlling for traditional and confounding risk factors. The model's theoretical framework is displayed on the screen. The dependent variable is the population percentage of cholesterol prevalence. The coefficients B1, B2, and B3 capture the independent effects of our water contaminants, while B4, B5, and B6 isolate the effects of our demographic confounders, age, obesity, and smoking. Epsilon represents the residual system error. This structure allowed me to calculate the specific coefficients in the model's overall R-squared value. Here is a look at the dataset. Table 1 shows the results of the regression analysis and indicate that obesity, P equals 0.008, is the strongest individual predictor on the model, while median age, P equals 0.021, is a very strong significant predictor, and PFOA and PFOS, P equals 0.048 and 0.049, are just below the standard 0.05 threshold, meaning they are statistically significant. The R-squared value indicates that the model explains 78% of the variance in the dependent variable. The central finding of this model is that even after accounting for major risk factors like obesity, smoking, and age, PFOA and PFOS exposure remains significantly associated with high cholesterol prevalence. This supports the conclusion that these compounds contribute independently to cardiovascular risk. Looking at the coefficients, the model predicts that for every 1 part per trillion increase in PFOA, there is an associated 0.2% increase in cholesterol prevalence. For every 1 part per trillion increase in PFOS, there is a 0.08% increase. While these shifts may seem small at the population level, they translate to a 1 to 3% higher community cholesterol rate. In public health, a 1% to 3% population-wide increase is a major cardiovascular risk factor and is highly significant and clinically meaningful. It is also essential to note the limitations of this study. First, my sample is restricted to a limited data set of 27 Ohio cities. Second, I utilized ecological population level statistics rather than individual health records, meaning I cannot map specific individual exposures directly to personal medical outcomes. Third, municipal water reports represent limited monitoring points, which may not capture household level tap variations. Finally, my model could not account for individual confounding factors such as diet, genetics, or regional medication use. These limitations should be factored into the interpretation of the results. In conclusion, the multivariable linear regression model supports the finding that PFOA and PFOS exposure in drinking water is associated with higher population level cholesterol prevalence, even after controlling for obesity, smoking, and age. These findings carry important implications for public policy and clinical practice. Because PFAS chemicals accumulate and do not disappear, my results highlight the need for strict water standards, continuous monitoring, and the implementation of advanced water treatment technologies to filter water down below to the recommended 4 parts per trillion level. Furthermore, the relevance of these findings extends beyond Ohio to broader public health practices. Currently, the healthcare providers rarely assess environmental exposure history when evaluating cardiovascular risk. Incorporating questions regarding drinking water sources and potential PFAS exposure into standard clinical screenings could allow for earlier identification of at-risk populations, ultimately improving long-term cardiovascular outcomes. This is especially important for any population-level finding. Thank William Mason High School and the public health organizations that maintain the open access data vital to student research. Thank you for your time.